Okay, welcome everybody again to another episode of the ideaXme show that profiles the humans behind the really big ideas that are shaping our world, inspiring future creators, and for all those that like really great stories. I'm Ira Pastor, your health, aging, and longevity ambassador along for this journey. So today is a very exciting show because aside from having a great guest joining us, we're also going to be moving into some areas within the so-called biologic hierarchy of life, disease, and aging that we have not talked that much about yet. One of these has to do with the general topic of biologic form. That is sort of the unique biochemical and biophysical dynamics that dictate these higher order things like size, shape, polarity, movement across our cells, tissues, and organs, which are involved both in health maintenance, but also disease transitions. Secondly, we're also going to be talking about some very interesting discoveries in the area of biologic robustness and resilience. That is, the respective ability of our cells, tissues, and organs to resist negative changes as well as bounce back from them when they do occur. Once again, how these processes can support our health as well as be hijacked in disease transitions. And during this discussion, we're going to learn a wonderful new word in the process, as we occasionally do on this show, this time Anastasis, which represents a novel biologic phenomena discovered and named by our guest about the ability of cells at the very border of life and death to, in essence, resurrect themselves. So with that, I'd like to introduce our guest, Dr. Denise Montell. She is the Robert and Patricia Duggan Chair in Mathematical Life and Physical Sciences at UC Santa Barbara's Department of Molecular, Cellular, and Developmental Biology. Dr. Montell earned her BS in biochemistry and cell biology at University of California, PhD in neuroscience at Stanford. As an American Cancer Society postdoc fellow at the Carnegie Institution, she developed entirely new methods for studying cell motility in vivo using combinations of cell biology and molecular genetic approaches. She became a full professor in 2002 at Johns Hopkins School of Medicine, where she served as director of the graduate program in biologic chemistry for 10 years and was the founding director of the Center for Cell Dynamics, an interdisciplinary group of investigators from 8 different departments who shared a common interest in observing and manipulating biochemical events and cellular behavior in real time in living cells, tissues, and organisms. She currently serves on the advisory councils of both the American Cancer Society and the National Institutes of General Medical Sciences. Recently joined the faculty at MCDB, UC Santa Barbara, to pursue her interests in both research, teaching, and program building. Dr. Montell, thank you very much for joining us today on the show.
Thanks for your interest in our work.
Absolutely. So we typically just start this show off by giving our guests the floor just to hear more about you, your background, where you grew up, how you got interested in science, how you got interested in health, and ultimately the pathway that brought you to this extremely interesting intersection of these disciplines that we don't usually hear as much about in the world of biotech.
I grew up in New Orleans, which is a town that people associate with a lot of things, but usually not so much science. Mm-hmm. And it's a fun place, and it's a place where I kind of learned to work hard and play hard, and that's kind of been a theme. My parents were both professors of microbiology at Tulane Medical School, mm-hmm. So I kind of literally grew up in laboratories and playing with stuff in the lab, and so I guess it's not too surprising that I ended up going into the sort of family business of science. They were both interested in microbiology and studying microorganisms, and I got interested in neuroscience and the brain when I went to mm-hmm. college. So I went to college at UC San Diego, and I explored other, sort of intellectual disciplines and things like that. But I was interested in philosophy, but in philosophy, people are still asking the same questions they asked 5,000 years ago and not getting too much closer to answers. So I appreciated that in science, we ask maybe sometimes smaller questions, but we can get some pretty hard answers. And so that was what really attracted me, the idea that you could explain seemingly complex phenomena with simple, elegant explanations. So, and the electrical activity in your brain comes down to the movement of a few ions down their concentration gradients. That's mm-hmm. just so magical to be able to explain things that way. So I got interested in neuroscience. I had a very charismatic professor, Nick Spitzer. He's still there at UCSD, and he really got me interested in that subject. So then I did my PhD at Stanford in neuroscience, and really got kind of interested at that time. The biggest, or one of the biggest mysteries, I was interested in consciousness. Okay, I wanted to understand the physical basis of a conscious thought.
That's a big mystery.
It was 30 years ago. It was an even bigger mystery, I gotta say. And so, well, no, 40 years ago. And so, you know, I went to this graduate program in neuroscience, and yeah, we weren't going to be able to get at that issue directly, but what we did think we could start to understand is how the brain wires itself up during embryonic development. Because, you know, the human brain is filled with billions of neurons and trillions of synapses, and that's sort of a problem too complicated to wrap your head around. So we started studying simpler animals to try to get at this, assuming that in evolution, you know, probably how you wire brain started out by wiring a simple brain, and then maybe the same principles would apply to more complicated animals. And that's sort of a theme throughout my career, is that, and I think it holds up pretty well for a lot of different biological processes, that evolution invented them early, they're ancient, and they're present in many different organisms. So we were studying fruit flies, and my graduate advisor started out studying grasshoppers and moved on to fruit flies. And we could, where there were, you know, more countable numbers of cells, and the wiring diagram was simpler. And so then we worked on trying to figure that out, identify molecules involved in that. And I realized at the time, even that was still a complicated problem, and that we wanted to understand just fundamentally how cells move around. How do they move? How do they know where to go? Because cells have to move to build an embryo, to build a brain, to heal a wound, to make the immune system function. Cell movement is really a fundamental property of animal life, a defining property really almost of animal life. So
absolutely.
Yeah, so from that exploration into neuroscience, then as a postdoctoral fellow, I decided to simplify the problem even more. Instead of trying to study motility in the complicated setting of the brain, we decided to study it in a much simplified system in the fruit fly ovary, where we really could count and see every single cell and every part of the cell. And so there was a lot greater opportunity to really understand mechanistically what was going on. Yeah, and then from there, from the postdoctoral studies, I started my lab at Johns Hopkins School of Medicine, and we've been exploring. Now, the way I think of it is, if I were to define what we work on in my lab, is we identify kind of quirky, unusual, understudied cell behaviors, and then try to figure out what are they good for in life, and then how do they happen and how do they work, so we can manipulate these cellular behaviors. And one of those, as you alluded to, is this ability of cells to recover from the brink of death. And that you know, we discovered that cells can do this. You know, every single day, every one of the trillions of cells in your body makes an active decision whether to live or to die. And you might think, wow, I want my cells to live. You know, I want to live, I want my cells to live. But it's actually really important that some of them die. It's really important that, you know, we generate every day lots of immune cells that have the potential to attack our own tissues. And the ones that are going to attack foreign invaders, we need to keep, but the ones that are going to attack our own tissues, we need to kill off.
Sure.
And they kill themselves off through this cell suicide program known as apoptosis. And so other types of cells that it's really important that they die would be, you know, sort of old and damaged cells. So our immune cells don't last our whole life. They're produced and turned over every day. Or the cells lining your gut, you know, they're exposed to a lot of harmful things in the environment, and they have to turn over, and have the old ones die, and then new ones are produced. So those are different situations where it's important for some cells to die. And then if you have a cell that acquires, let's say you go out in the sun and you get exposed to UV light, and you acquire a mutation in the DNA in a cell, if the cells are damaged in that way, it's again kind of important that they die, so that we don't develop cancer every time we get exposed to something harmful. So incipient cancer cells, old or damaged cells, autoreactive T cells, all of these kinds of cells need to die. But then, of course, we want our cardiomyocytes in our heart to last our whole life. We want the neurons in our brain to last our whole life, and a lot of cells we want our muscle cells to last a long time. And so these life and death decisions at the cellular level are really critical for our health and well-being. And it turns out that cells can commit suicide or not, and they have to decide and whether or not to do that.
Mm-hmm.
And so when they're exposed to some kind of toxin or radiation or something harmful, cells will often go ahead, if they're damaged enough, commit suicide. But we discovered that if we treat them with a toxin that will induce them to initiate the cell suicide process, if we wash that toxin away,
Mm-hmm.
then the cells can bounce back and recover. And that could be good news if that's a cell that you want to keep in your body, but that could be bad news if it's an incipient tumor cell or an autoreactive T cell or something like that. So, so there are positive and negative implications of that, and we'd really like to understand how that process works so that we can harness it. That's one quirky cell behavior that we study. We study other ones, cells cannibalizing other cells, cells moving around the body in different types of groups, collectively behaving like a flock of birds, but flock of cells.
Mm-hmm.
So there's a bunch of different kind of behaviors that we study.
It's extremely fascinating, and if we could stay on the topic of anastasis for a little bit, I mean, it's a truly amazing discovery in the sense of, there are these points of no return that the scientific community has thought of for so long, and here you come along and wow, it's you have the ability to actually reverse beyond a point of no return. And as I said, it has these positive consequences in terms of potential learnings for regenerative medicine, at the same time, chemo resistance and cancer and so forth. Could you tell us a little bit? I mean, I know this is very early stage stuff and nothing confidential, of course, but things that you're learning about gene expression and morphodynamics and so forth of these cells, because, a lot of on this show in the past, as we talk about sort of the area of aging. We talk a lot about, as you mentioned, the damage, senescence, apoptosis, death cascade, and now you're flipping things around here. Is it anti-senescent genes? Are they somehow embryonic type genes that are trying to rebirth the cell itself? What are some of the interesting things you're finding out about this process?
We thought that obviously those are all reasonable proposals, that maybe this would revert cells to more of a stem-like quality. We don't actually see that so much. So what we did was we took some cells, HeLa cells, in a dish and exposed them. We collected some of them without treating them at all, and then we took other cells and we exposed them to a toxin that would induce apoptosis. And we'd leave them there for 3 hours, and they would shrivel up and look terrible and be on the brink. And if we were to leave them in that condition, they would die. But we'd collect some that were on the brink of death, and then we'd take other cells and expose them to that toxin, and then wash the toxin away and allow them to recover. And we would collect them at different time points during the recovery process, and then profile all the gene expression that was going on in the cells at these different times and compare it and ask, what does this process look like? And we a priori didn't know anything about what that process would look like, but we could see from the profile. First of all, the first thing we learned that was very clear, it fell right out of the data, was that this was a two-step process. That the first, we collected cells 1 hour, 2 hours, 3 hours, 4 hours, 8 hours, 12 hours as they're recovering. 1, 2, 3, 4 hours all look really similar. And 8 and 12 hours look really different from that. And they're not the same after 12 hours as they were before they started. So they've recovered, but they haven't recovered to precisely the same, indicating that there might be some kind of epigenetic memory of this near-death experience. They might have been permanently affected in some way by this near-death experience, which might have, so for example, you could imagine that they might be more resilient to a subsequent stress. Or they might be less resilient to a subsequent stress. So, okay, so it might have changed them in some way. And then that we could see that, and then we could see these two phases. The early phase, what happens immediately when the cells start to recover is that they immediately start to express lots of other transcription factors. So they turn on this whole transcription program really quickly. And then they also start proliferating, so they start dividing. And in the late phase, they kind of pause proliferation and they become migratory, okay? So what this looked like to us was a wound healing process. If you're a cell, you're in a tissue, you've just been exposed to some horrible condition, you almost died, but maybe your neighbors did die, and some of the neighbors did die. What would you want to do to repair that tissue? You would proliferate, you would secrete some angiogenic factors to call in blood vessels for nourishment, you would proliferate and divide to fill in the gaps, and then you'd migrate to seal off the wound, you know? And so to us, this program that we saw in the pattern of gene expression looked like a wound healing process. And that was interesting because it made us think, well, that makes sense. Maybe this is the normal function of anastasis, is to allow a tissue to repair itself after a severe but temporary sort of exposure to something harmful. Okay. So if you and your listeners are up on apoptosis, then you think as well, okay, this is a way to eliminate damaged or dangerous cells that might turn into cancers and stuff. So why would an organism evolve the ability to reverse that? If it's doing you good, why would you reverse that process? And so what we thought of and what we saw and seemed to bear out in these results was that it might have evolved in order to limit the permanent tissue damage that you would have in response to a severe but temporary kind of harm, whether it was a chemical exposure or radiation exposure or what. So if you're exposed to a big burst of radiation or a poison, you wouldn't want all your cells to commit apoptosis at the same time. That would not be beneficial.
Right. Makes sense.
So we saw in the molecular profile, we saw evidence for that. And then some of our work on anastasis we do in human cells and in the lab, and some of the work we do in Drosophila, in the fruit fly, because like I said, some of these very fundamental cellular behaviors are ancient and deeply conserved across organisms. And we're seeing that bear out in work that we're getting ready to publish in the fly. We can see that if we expose the animals to radiation or other types of stress, that cells will undergo anastasis and contribute to the repair and recovery of the tissue.
Fascinating. Very fascinating. So, you know, one other, you know, fascinating things, but, you know, one other, you know, major component that your lab is targeting has to do with organisms like Drosophila to enter these stress-resistant or sort of hibernation type states, and you know, it's sort of an interesting topic in the last couple of years. There's been a lot of sort of activity on the tardigrade front with, you know, how some of these really ultra stress resistant organisms can dehydrate themselves and put themselves into long-term stasis to, you know, to resist all sorts of crazy things. I know the military is looking at ways that we can use this knowledge to, you know, preserve soldiers in that golden hour where they could bleed to death and so forth. You know, you've been doing a lot of analysis on Drosophila in this sense. You know, can you tell us a little bit about sort of what you're learning on that front, especially it may pertain to this, you know, stem cell maintenance and the niches, and ultimately, you know, going back to the topic of aging, which everyone's so interested about with regard to the show.
So this is a pretty new project in the lab. And it's kind of fun because I've been working on Drosophila for, you know, decades, and thousands and thousands of people across the world, believe it or not, who do. But most people who work on Drosophila don't actually even realize that fruit flies can do this. Okay, so it turns out that fruit flies normally like the same kinds of temperatures that we do. They grow optimally between 18 and 25 degrees centigrade. And that's, we like to say that's the Santa Barbara climate, you know. But it turns out that if they are born, we call it eclosing for a fruit fly, but if they're born, come out as adults in 10 degrees, which would be like 50 degrees-ish Fahrenheit, then they think it's late fall, and they enter this state called adult reproductive diapause, which is kind of one of these dormancy or quiescence type of states where they don't mate, they don't eat as much, they don't reproduce, and they don't age. So and they can stay there for weeks and weeks and weeks, and then when you put them at the 18 or 25 degrees, then they can resume their non-diapause activities, and they can mate and they can reproduce and so on. So we got interested in this. I heard about it first from one of the elder statesmen of, sort of fruit fly biology. I was at some conference and it was one of the few people who really knew about it and mentioned it over lunch. And so I went back to the lab and I'm like, I'm going to study that process. It took us a while to set up our 10-degree room and like do all this stuff. But anyway, so we've been studying that, and one of the things that we found right away, it turned out that, some people had studied it and published papers on it, but we were fascinated by the idea that they could preserve fertility, female fertility in particular. They could preserve this female fertility for much longer than a non-diaposing animal. And since that's something that typically declines with age in lots of different organisms, we were interested in understanding how they do that. So the people who had studied diapause in the past were more kind of physiology type of people, and we study reproduction and stem cells in the ovary, and so we have the ability to look at much greater resolution, higher resolution at what's going on with the stem cells. And we know more now than people used to know about the normal biology of stem cells in the ovary. So we started looking at that, and we've discovered that and we haven't published this yet, but we're getting ready to write it up, is that normally the stem cells that are going to result in the production of eggs sit at the tip of the ovary. They sit in this little niche that's protecting them and maintaining their stem cell behavior. And the niche is composed of some other cells that are secreting some goodies that the germline stem cells are receiving. And what happens during diapause is that actually the germline stem cells simply kind of turn their back on the niche. They kind of unplug and disconnect from the cells in the niche, and they hang out there. They don't differentiate, so they don't turn into differentiated cells. They don't progress in their development, but they also don't listen to the signals they would normally be getting from the niche that's telling them to divide and produce more progeny and all of this sort of stuff. So they have basically kind of found this this way to hang out, to chill out, so to speak, and not really act like a stem cell, but also not really differentiate either. So they find a way to maintain this in-between state. And then when we put the flies back into an 18-degree or a 25-degree temperature, the cells turn back around and plug back into the niche. And at the lower temperature, the niche has been maintained better. We haven't lost cells from the niche as we would during normal normal life at 25 degrees. So there are more niche cells. The stem cells haven't been dividing, so they've maintained their replicative potential because they haven't been dividing all this time and producing progeny. They've probably incurred less damage as a result of not doing that cell division. And so, this question is, how is it that they unplug and replug in? Like, how exactly does that work? And that's what we're working to try to figure out. But we had to sort of look at them and see what they were doing just to even know what question to ask there.
Absolutely. I haven't played around with Drosophila too much, but I do. Yeah, wouldn't know any of that about them, but that's.
But there are a lot of, so the first stem cell niche ever discovered was right here. This one, this germline stem cell, and this were the first ones that were ever sort of described at a cellular and sort of molecular level, and so we think that this principle might, well, easily apply to other types of stem cells that, if you can slow down their rate of division over time so they don't accumulate damage, this is a useful thing.
So one other topic I wanted to go, and clearly it's one of the major area that you work in, and this topic of form. One that sort of I've always been extremely fascinated by, especially as it pertains to biologic systems, not just because, as mentioned before, these things like size and shape and movement, these are not phenomena that are easily connected to, this specific gene or that protein, but really involve these complex interactions between biochemical and biophysical networks. Your lab is very focused on major aspects of form, namely movements and motility, which, as you point out, if it doesn't occur properly, embryos don't develop into fetuses, and wounds don't heal, and the nervous system doesn't connect and do all the things it's supposed to do. Very important for life, but also, these things can be hijacked to, once again, for metastasis and so forth. Can you tell us a little bit more about, sort of things you're working on in this space? And then also, I realize that there's probably, when it comes to form, you move beyond sort of the traditional, as I said, the gene and the protein, but you're working with membrane potentials and forces of mechanotransduction and all sorts of higher order dynamics that we don't normally, once again, in sort of in the pharma industry hear too much about, but are just extremely important for the big picture, let's say, of why stuff works the way it does. So
yeah, I think in the maybe biotech arena where this is probably most important is when you're thinking about regenerating tissues.
Sure.
Right? So you can have a stem cell and it can make all its progeny, but if they don't organize themselves in the proper way, then you're not going to have a functional organ or tissue. And I think the biggest challenge in regenerative medicine, from my perspective right now, is once you've made the cell that you want to replace some damaged or diseased or missing part in your body, how are you going to get that thing to integrate into the environment that's already there? So we have to understand the principles by which cells normally self-organize into organs and tissues, so that we can have any chance of being able to repair damaged or diseased tissues with the beautiful cells that we can produce in culture from stem cells. So, that seems to be going really well. We can grow stem cells, we can turn them into all kinds of different cells, but how do we get them back in the body, in the right place, connected to the right partners, and doing their job? So this is where I think, this is a really interesting problem. It's also just an interesting fundamental science problem, how do cells organize? And so it seems more complicated than, this gene encodes this protein, which does this function, and when that function's missing, here's the very precise defect that you get, like sickle cell anemia or something, right?
Right.
Or even cystic fibrosis or whatever, there's like gene, protein, function, and then disease. It seems more complicated than that, but and I've been thinking about this for a long time, and I've been writing about it a little bit. It's not infinitely complicated. We actually can boil down all the diverse cell shapes and organizations that we see in the body. And there are hundreds of types of cells. They can acquire many different types of shapes, and they can adhere to one another or not. They can adhere to a substrate or not. They can be tall and skinny, or they can be short and chubby, or whatever. So, and it seems very, very complicated. But in fact, I think that it's definitely not infinitely complicated. I think that biology generally, bear with me while I reason through this with you,
Sure,
biology generally builds complexity out of simple modular units.
sure.
Okay, so DNA, 4 base pairs, lots of complexity in genetic information.
Absolutely.
20 amino acids, lots of complexity in the protein space in terms of, tens of thousands of different proteins and potentially, many more. Biology builds complexity out of simple modular components.
Mm-hmm.
Think about your anatomy is also built out of simple modular components. Your skeleton, like each segment of your body has similarities to the other one and then differences. So we have modules and we diversify them, but they're simple modules and we build complexity from that. So my thinking around this area of self-organization of cells and cell morphology is similarly that all that apparent complexity is really built out of really just 4 simple mechanical properties that cells have. Okay, they can adhere to one another, they can adhere to a substrate, they can contract with myosin, with the sort of muscle type proteins that every cell has. They can contract and make themselves more round and small, or they can protrude, they can polymerize actin, and they can extend outward. And if you tune those properties, you make them more adhesive and less contractile, you'll get very pronounced shape changes and a propensity for the cells to associate with one another or to be loners. And so I think that there are principles here. There are these modules. The modules are the mechanical properties of cells. And as you said, it's maybe not as simple as one gene, one protein, one property, but there are some important genes and proteins that are kind of the master regulators of certain properties. So myosin, this protein that's most famous for making your muscles contract. Every cell has a version of it that allows an individual cell to contract.
Okay.
And that is one protein, one property is this contractility property. Or cell-cell adhesion, we have lots of families of cell-cell adhesion molecules. So there are molecules that make cells want to stick together. And so these properties that can be combined in different ways are they are encoded by molecules. It's just not as simple as one molecule, one property, but it's not that bad, actually. So we think you can tune these properties by regulating molecules, and this will give you a lot of diversity in morphology and organization of cells.
Simplifying the complex. I like it.
Well, yeah, that's that's what attracted me to science, was the ability to explain complicated things by simple mechanisms. Unfortunately, the ones I study aren't that simple.
Oh, wonderful. Well, we're getting to the part of the show where we talked a lot about science. It's completely eye-opening and fascinating, and the work you're doing, and it is a thank you for moving this area of science forward as you are. And I can see once again all of the amazing connections to regenerative medicine, to oncology, to neurology. It's an amazing basket of potential. But I have to go to the point now of the show where we move from the science now into the science fiction. And I have to pose the following question to you. If I was to give Dr. Denise Montell a time machine, my time machine that I have sitting over here, it's not peer reviewed yet, but it's functional, and I allow you to travel anywhere you want to go, who would you like to go visit? You don't have to be a scientist, be an artist, philosopher, whatever. And you can go visit them and talk to them for a while about something. Who would that be for Dr. Denise Montell?
Oh my gosh, that's easy.
Okay.
It would be Aristotle,
Ooh.
Without question, because he's all of the things you just described,
Right.
An artist, scientist, philosopher, and all the rest. But it is absolutely amazing how many things he got right.
Sure.
Incredibly limited technology available to him at the time, and all these diverse areas of thought where he was just so brilliant. And what I would love to do is have some time with Aristotle to bring him up to date on like what we're doing now. And he didn't get everything right, but he got a lot of things pretty darn close to right. Tremendous insights. And so if I could just bring him up to date on what we know, which probably wouldn't take too long because he's a quick study, I think, then I would love to hear what Aristotle's, what he would see in what's available to us now that the rest of us are missing, because I'm sure he would have some really tremendous insights.
So, outstanding.
So get that time machine up and running, because I really would like to do that.
It's the next thing I have to do. Well, it's really it's been a major honor having you here, and really want to thank you for joining us and taking the time out. Obviously, you have so much going on, but for everybody once again watching and listening, it's been Dr. Denise Montell from the Mathematical, Life, and Physical Sciences at the UC Santa Barbara Department of Molecular, Cellular, Developmental Biology, doing truly amazing things in the study of biologic form, the discovery of a fascinating phenomenon known as anastasis, as well as amazing new work that you're going into in regard to stress-resistant states, a completely amazing amount of research by one laboratory that is really, as we like to say in the show, really moving the human story forward. And not just, thank you for being on the show, but a thank you to everything that you do and that you're doing for humankind and the human situation. This is really fascinating, wonderful.
Thank you for having me. And I really enjoy looking at your podcasts. They're really fun.